Hi, and welcome to my talk, Quant it all and Quant it now, answers from only microliters with Uncle and Stunner. This talk is about all the data you can squeeze from just a droplet of sample. In this case, protein quantification, aggregation, and stability information on two platforms that each use less than 10 microliters of sample. When I look at this field of sheep trimming grass all to the same length, I'm reminded of traditional protein quantification. That's because quantifying protein concentration can feel like quite a dull activity, when all you're trying to do is get everything to the same concentration, and the available technologies all seem to be lots of slow, one at a time, or only kind of accurate options. Thankfully, Stunner is none of that. Stunner combines two different technologies in one instrument for the first time to enable answers on quantification and aggregation all at once. 2 microliters of sample gets the concentration of your protein samples while making sure that you're measuring monodispersed proteins, not a bunch of aggregates. This is done for up to 96 samples per run in a plate-based format. So from one read, Stunner can gather UV-Vis absorbance information to determine concentration and DLS information to determine size and size distribution. Stunner can measure proteins across a broad range of low and high concentrations. It uses a microfluidic circuit with 2 fixed path lengths to cover a wide dynamic range of 0.03 to 275 OD. The unique microfluidic circuits molded into Stunner plates ensure that there's no cross-contamination from read to read or evaporation, and the fixed path lengths mean you know exactly what you're measuring and give extremely good precision. How does Stunner do this? Well, when samples are deposited into the input well, they are then drawn to the microfluidic reservoir of the circuit. A sample can be left in the microfluidic reservoir for up to 2 hours without detectable evaporation. During a read, Stunner pumps a sample from the microfluidic reservoir into the microcuvettes. Stunner circuits have 2 microcuvettes in each circuit with 0.1 and 0.7 millimeter path lengths. The 2 different sized microcuvettes make the wide dynamic range possible. The 0.1 millimeter cuvette provides a short enough path length to enable high maximum detectable absorbance, while the 0.7 millimeter path length enables reads at low concentrations. And with that technology behind it, Stunner delivers amazing results. Accuracy within 2% and precision within 1% from 1 to 200 OD. Let's check out a few examples of exactly how great those results are. To demonstrate Stunner's accuracy, uracil and tryptophan NIST standards, known as Standard Reference Material 2082, were measured. Here on the y-axis we're showing percent bias, which is the absolute value of the percent difference between an expected and a measured result. For these materials, all the results calculated based on 8 measured absorbances in each of 5 instruments were within 1.65% of the expected values. Further, the mean shown by the x and the median shown by the green line were within 1.25% of the expected values. Pretty spot on. If we want to take a look at determining concentrations of a molecule, maybe more representative of the real world, we used NISMAB. NISMAB is a gold standard IgG that's known as a common standard for therapeutic protein characterization. According to standards defined by the USP, analytical methods for determining drug concentrations should be within 2% of the known concentration, plus or minus the manufacturer's uncertainty. The NISMAB manufacturer's uncertainty is about 0.4% of the reference value of 10 mg per mil. So an analytical quantification method should be within 2.4% of the reference concentration. All measurements in the 5 tested instruments shown here were within 1.25% of that 10 mg per mil reference concentration of NISMAB, as shown by the whiskers of the box and whisker plot. Mean and median values of the 40 measurements were within 0.6% of the reference values. If we want to take a look at an even more real-world example, here on the left, we're looking at the full spectrum absorbance UV-Vis signal of an IgG dilution in a PBS buffer. As we further dilute the signal, dilute the sample, you can watch as the signal goes down. On the right side of the slide, we're looking at the linearity of that same sample, where across the x-axis we're looking at concentration, known concentration, and in the y-axis we're looking at the A280, which is the measured absorbance value at 280 nanometers. You can see the linearity of this result is 0.9999. If we want to then quantify this sample of known concentration versus measured concentration based on that A280 and the known extinction value for this protein, we can see that it gives a slope very, very close to 1 and an R-squared value again of 0.9999. So we know the results are very, very linear. If you look at the data in tabular format, it makes it a little bit easier to look at accuracy and precision, where we notice that the average concentration is going to be within 2% of the known concentration, and the CV is within 1% of all values. So that's where we get the 2% and 1% values for accuracy and precision. And if you want to show off this accuracy in your lab, we also make a fundamentals kit, which is a set of certified tryptophan standards across an OD range of 20 to 225, which is equivalent to a 20, 40, 80, 120, 160 mg per mil concentration of IgG. When we run results from when we take a look at the results from the fundamentals kit, we can again look at target versus measured concentration and see results with slopes very, very close to 1 and R-squared values above 0.9999. And if we take a look at the right, we can see how the absorbance of tryptophan behaves across the full spectrum of measured absorbance. And again, looking at precision, every value here is performing better than a 1% precision specification, with, in fact, the highest concentration only having a RSD of 0.52%. And all of this is automation compatible too, because Stunner talks to robots, so you can simply set it up with your automation system and forget about loading and pipetting and measuring all of your samples. And beyond that, it's also downstream ready with 21 CFR Part 11 software, performance verification, and across 6 different measurements, including absorbance accuracy and precision, photometric linearity, wavelength accuracy and resolution, and stray light, which means that you'll be in compliance with both the USP and the European Pharmacopoeia standards for UV-Vis instrument. And so that's how Stunner supplies speed, accuracy, and even maybe a little bit of fun to protein quantification. So that brings us to UNCLE, which is the multimodal all-in-one biologic stability platform that has the right number of options to investigate and solve protein problems. This talk will look a little bit at how UNCLE combines 3 different detection methods, which is full spectrum fluorescence, static light scattering, and dynamic light scattering, across 12 applications using only 9 microliters of sample to help you understand protein stability. So one point of this talk is that if you only look at things one way, it can be unclear exactly what you're looking at until you take a different approach to see what's right in front of you. So how does UNCLE help you do that? Well, by combining 3 different detection modes in one instrument, it gives you the flexibility to really understand your sample that you're investigating. So the fluorescence is a full spectrum fluorescence read so that you get the full picture of your protein fluorescence, whether that's intrinsic protein fluorescence done without label or whether that's done with a fluorescent dye. SLS, or static light scattering, is going to give you extremely sensitive detection of aggregation that's going to give you answers at the same time as your fluorescence data. They'll be very, very sensitive and rapid and will line up to melting temperature events. And then DLS, or dynamic light scattering, is again going to let you crank up the detail and see size and size distribution data on your samples. And all of this is done with temperature control, so it can be, you can take a quick snapshot at room temperature, or you can run a thermal ramp and use these 3 detection methods to get information throughout that experiment. So the unique features of the UNCLE all start from the UNI, which is an array of 16 quartz microcuvettes that hold 9 microliters of sample and that can be clipped inside of between silicone seals inside of a blue frame. This is what enables us to gather all that information on only 9 microliters of sample. When we're studying samples, it's important to also understand how proteins fluoresce. So in this case, we're showing the excitation and emission behavior of tryptophan, tyrosine, and phenylalanine amino acids. The idea being that if we're exciting these amino acids with a UV laser, then we're going to be seeing them fluoresce, and when they're present in a protein, they're what gives total intrinsic protein fluorescence. If we look at that across a full spectrum read on the UNCLE, what we'll see is this. So going from the top to the bottom is each read of fluorescence on the uncle done at a different temperature, starting from room temperature going all the way up to 95 degrees C. We'll take the area under that curve and find the center of mass wavelength, which we'll call the BCM wavelength. That's how we'll track any shift in this fluorescence spectra, which will be information that helps us determine protein unfolding. Static light scattering can be seen on this slide at 266 nanometers, which is where our UV laser excites the protein and then bounces back out and gives us information on aggregation. So here's what a thermal stability experiment looks like. In blue, we're tracking that BCM wavelength, which is that center of mass wavelength that tells us information about protein unfolding. And in green, we're tracking static light scattering, which is information on aggregation. So in blue, as the protein's fluorescence shifts to the right, or red shifts, we'll note that by looking at inflection points in that curve, which we've labeled here as TM1 and TM2. If we want to track aggregation at the same time, we can look at the SLS data and understand exactly when proteins start to aggregate, which is going to line up with, in this case, melting temperature 2, and understanding both of those melting temperatures at the same time is going to be very important to understanding when exactly your aggregation event occurs. And in terms of when that can occur, well, you always want to check and see when your static light scattering events line up with. In this case, they can line up with the first melting temperature, or they can line up with the second melting temperature, as seen in trastuzumab, or they can actually occur before any melting temperature. So that's why it's key to understand exactly when aggregation starts, because that should line up either before melting temperature or simultaneously with an aggregation, a melting temperature. And using this kind of information on uncle, we can quickly screen not only proteins, as you just saw, but also formulation conditions to understand unfolding and aggregation very quickly across a panel of formulations. Okay, so let's take a look at an example. Here we have a protein with one native tyrosine that we're interested in. If we take that and compare it to a mutant where we added a second tyrosine, we want to study and see if this changes the protein stability of that protein. And if we run the sample on uncle, we can analyze it two ways and see kind of an interesting story. So on the left, we're studying the BCM wavelength, again, that center of mass wavelength that takes in the holistic picture of the whole protein intrinsic fluorescence, and see that a one tyrosine protein has a relatively flat signal, while a two tyrosine protein does show an increasing signal by BCM, indicating a redshift in your protein. However, if we study just the ratio with this protein, the 350 to 330 nanometer ratio is typical, the two tyrosine protein now is the one that has a flat signal, and a one tyrosine protein actually implies that a blue shift is occurring, which is something not seen with the BCM signal. So what's going on? Well, with uncle, we can actually investigate it further and understand those results. So if we look at the signal from the protein at the top, the one tyrosine protein, we can see that the two kind of characteristic markers if we compare it to the two tyrosine protein. First, if we're looking at the green arrow, the one tyrosine protein at the top actually shows a stronger tyrosine peak than the two tyrosine protein at the bottom. So the amplitude around 300 nanometers is higher. So that's kind of a first counterintuitive result. Secondly, shown with the blue arrow, the two tyrosine protein appears to show more of a redshift than the one tyrosine protein. So this will be important to remember. We actually take this data and look at where the 350 and 330 nanometer ratios hit on this fluorescence, we can understand how those would miss both the tyrosine signal peak shown on the shoulder and miss that red shift that's more shown by the blue arrow in the two tyrosine protein. If we take this data and graph it in 3 dimensions using something like Excel, we can see, again, where that green arrow shows a stronger tyrosine signal in the one tyrosine protein. And if we're looking at the top-down view in the bottom right here, we can understand where that two tyrosine shift starts occurring and see that red shift by looking at the topographic map here. And if we want to start to understand why that shift is occurring, we can actually start to run some control samples. So here we're comparing the result from that two tyrosine mutant against the result where we've spiked in an IgG sample with some dityrosine, and understand that that signal is remarkably similar, and so that signal coming from the two tyrosine mutant probably indicates that tyrosine is cross-linking and forming a dityrosine degradation product. So with UNCLE, you're able to see the difference, investigate, and ID the cause there. And beyond that, we can also use UNCLE's full spectrum fluorescence to use dyes to understand your protein further. So in this case, we're using the 473 nanometer blue laser. That's the second laser on UNCLE. That'll also give you SLS data at 473 nanometers. And this will be exciting any kind of extrinsic dye that you've added that'll be fluorescing in the 500 to 650 nanometer region. So in this case, we're using it to study a protein that actually does not have native tryptophan or tyrosines. So all we do is simply add some cypro orange, and we're able to use the blue laser on the UNCLE to excite cypro orange fluorescence. That's going to give us information about protein unfolding. And if we want to take a look at even more interesting samples, such as AAV, where we have both protein unfolding and DNA to be concerned with, we can study both stability pathways for AAV, where we have protein unfolding and capsid disruption shown on the top. And actually, we have a second pathway that's exclusive to DNA, where by adding cybergold dye, we can detect DNA ejection occurring from relatively intact capsids that don't show any signal from protein changes, but instead only show signal as cybergold dye binds to DNA, leaving capsids. And so, for example, the result from this kind of experiment looks like this, where we have green fluorescence data, starting at kind of a baseline for however much free DNA we have in a sample, a decreasing signal because there's a relationship between cybergold fluorescence and temperature. And then the headline, it starts at about 50 degrees Celsius and continues through an inflection point at about 58 degrees Celsius, where cybergold is showing you that DNA is releasing from your capsids and is now available to bind to cybergold and fluoresce. This DNA ejection continues to occur until about 75 degrees Celsius, where protein aggregation starts to happen, which is a result of unfolding of the capsid proteins. So this is important to note because here with one experiment, we've seen both DNA release and capsid aggregation, and it's telling us that DNA release from AAV capsids is occurring about 20 or 25 degrees before any kind of protein unfolding result. If we want to use DLS to look at our samples, we can also get really precise answers on Z-average diameter and PDI to understand size and size distribution in just a few seconds. And that can help us keep track of protein quality as it maybe starts as an aggregated sample shown in blue, and then deteriorates over time with more and more aggregates and more wider and wider Z-averages and PDIs. And we can do this kind of DLS read across the thermal ramp too, so we can simply track and quantify size change as it occurs in a sample. And so that's how only on UNCLE we can use fluorescence to see everything, investigate, and determine the cause of problems. And we can use static light scattering to understand exactly when protein aggregation occurs. And we can further use it to study things like B22 and KD. And then we can use dynamic light scattering to understand the quality of your protein sample and get quantitative answers on size and size distribution right away. So with that, we invite you to see things from all angles, including your protein stability problems. And now I'll ask, what questions do you have for me?